Hello, everyone, and welcome to this interview. I would like to welcome Professor David Hay from the University of Otago and Professor Andy Russo from the University of Iowa. I want to thank you first for accepting to join this interview. Today, we will be talking about their amazing session in the International Headache Society Congress on about Hope in the Horizon, the role of PACAP in migraine. So first of all, again, thank you for accepting to join this session. To begin with this interview, I would like to ask Professor David Hay about the splice variants of the PACAP receptors. I'm biased because I'm a pharmacologist myself. So I consider it's an extremely important topic in the field that has not been properly explored. So I would like to hear about the splice variants, about the opinion you have of these splice variants, and how can we harness them? How can we use these to our advantage?
Well, thanks for the opportunity to have further discussion on this following our session. Receptor splice variants are a fascinating topic overall. They are a hugely overlooked area in drug discovery and pharmacology. Any G protein coupled receptor that has more than 2 exons has the potential to be alternatively spliced. And so there are many examples of spliced receptors, but the PACAP receptor family appears to be particularly enriched in these receptors. So we have our PAC1N, which is PAC1 null, which is our kind of reference sequence for these receptors. And that's what we think about most of the time. And that's where our kind of canonical pharmacology comes from, with that potent PACAP response with a weaker VIP response. But then you have these variations in that sequence where small pieces of the coding sequence are removed kind of interchangeably between these receptors. So we have variations in the extracellular part of the receptor, which is important for binding to the ligands. We have the PAC1S receptor, short, and we have PAC1VS, very short. And these have different lengths of that key part of the receptor. And that does seem to result in some changes in the pharmacology of the receptor. For example, the PAC1S receptor, even though there's less receptor sequence, binds VIP with higher affinity. And that's kind of fascinating. And then we have our intracellular variants that may alter the signaling and regulation profile. And overall, we really just don't understand a huge amount about these receptors because they're quite tricky to study. It's tricky to understand the location of a receptor as it is. But when you have these variants, you have to be very precise in how you are identifying their expression in tissues. You're often having to rely on the RNA, which isn't necessarily telling us where the protein is expressed.
Okay, that's excellent. So when it comes to the variants, for example, how you just mentioned that it's hard to really study them. But for example, when it comes to the ligands that bind to them. So you just mentioned that BIP, for example, binds in a differential manner when it comes to the different variants. But what about the antagonists? Do we know if the antagonists also bind in a different manner to each splice variant?
So we have done some study of some of the variants. I mean, we have to appreciate that there are actually many of these variants, and we certainly haven't studied all of them. But the ones we have studied, we have tested some different antagonists, and they do appear to behave similarly, but the antagonism of these receptors is also very complicated, because the affinity of the antagonist depends on which agonist you use. And so it can be quite difficult to kind of unpack what's going on with this system.
Thank you so much. So going back with Professor Russo, for years we've heard like migraine is a severe disorder. Now we have this hope in the horizon when it comes to PACAP. What have we seen or what have you seen when it comes to animal studies? Do you see different responses to PACAP when compared to CGRP, or do you see similar responses?
First off, again, thank you for the opportunity to pontificate about my favorite peptide, CGRP, and its cousin, PACAP. I think one of the one take home I can give you up front is PACAP is more than just a CGRP wannabe. It's doing things differently than CGRP. It's similar. And so I think cousin is the right term for it. It's not identical twin. CGRP is very important in migraine. And we've been really, I think, fortunate as basic scientists to be part of this development of new drugs that are helping so many people with migraine, but they don't help everybody with migraine. And that really point out the fact that there's other players in migraine that are very important. And I think its cousin, PACAP, is going to be very important. Now, will the drugs work differently when they're targeting PACAP than those that work against CGRP? The answer to that, first off, we don't know for sure yet. This is all speculative, but I think it's pretty likely that they will work on a different group of people, maybe an overlapping group of people. Some people will respond to both CGRP drugs and PACAP drugs, is my prediction. And some will respond only to PACAP, or they respond only to CGRP. I'll circle back to that later in our conversation, but I'll just say up front, I think that PACAP is a really good contender to supplement and improve responses to CGRP. So people who don't respond completely to CGRP drugs, I think, will respond to PACAP-based drugs to improve the treatment efficacy.
Excellent. So with that in mind, for example, it's a bit of a charged question for both, but would you recommend then either define which patients are PACAP responsive and which patients are CGRP responsive, or should we just target both at the same time?
Go ahead.
Well, I think it's firstly tricky to know who is responsive to start with. I mean, if we look at what we've learned from the different CGRP therapies, I mean, there is evidence that a patient may not respond to one therapy, but may respond to a distinct one when in principle they're targeting the same pathway. So what's a true responder or non-responder anyway? So I think we're probably not in that position of being able to sort of segregate out. And then I guess in terms of targeting both, the majority of the evidence preclinically and now some clinically is pointing to them being independent pathways. So they're sort of working in a complementary way to modulate those biological systems. So if that's the case, then there is certainly the potential that you could combine and target both. I don't know what Andy thinks about that.
So I said I'd circle back, and Debbie just did a great job of drawing the circle for me. I think that our preclinical data support what Debbie just said, that CGRP and PACAP are acting independently. They don't depend on each other to have their efficacy in animal models of migraine. And we've shown that blocking CGRP signaling doesn't affect PACAP actions on mice and vice versa. And then the Masoud Ashina's lab has done a similar experiment now with people, showing that patients on arenomab, the monoclonal targeting the CGRP receptor, still can have a PACAP-induced migraine attack. So all the evidence from our preclinical studies, which I'd love to go into, but I won't, and the limited data from humans really points to the fact that targeting both in a patient will be more efficacious than targeting one or the other. Having said that, how should we start out first? I really think combinatorial therapy makes a lot of sense to do because migraine is multifactorial. It's not going to be just CGRP. Even though I think CGRP is the most important peptide on earth, there's other peptides out there, and it'll be more than just PACAP too. But from a safety issue, I think it'd be most prudent, and I'm not a clinician, so I just treat mice, but I think it'd be most prudent to start with just one at first and then see what the response is. And as Debbie said, it's been very difficult. At this meeting, they're talking about evidence that some people don't respond for 3 months to the CGRP treatments. And you start seeing an improvement at 6 months, and for some, it's not even until a year. So when do you decide that you're a non-responder or a responder is going to be tricky.
Thank you so much. So going back to that maybe PACAP is not the only one involved or CGRP is not the only one involved. What do you think the future holds? So, for example, when it comes to PACAP, is there a way we can sort of take advantage of these isoforms? Is there a way that we can create, for example, um, special molecules that only target one isoform or something like that, for example?
I mean, I certainly think that that's a possibility. I think we really have to understand more about which isoforms are actually involved. I mean, there's a lot of speculation around the Amgen PAC1 receptor antibody, and did that fail because it hit the wrong splice variant? We simply don't know. We would ideally learn more about that molecule, but I think what we can do is learn more about the biology. Let's work together to figure out which of these variants are actually important. And as far as we can, through the different species in a really cross-species translational way, if we understand that better, then certainly I think we've got a chance to selectively target or non-selectively target, whichever the better approach might be.
Okay, excellent. And what about?
I think that from my perspective, we're taking the right strategy with PACAP by focusing on the ligand at first. So let's do the proof of principle and show that that will help patients. And then, much as with the CGRP field, the monoclonal antibodies against the ligand really, I think, opened the door to really go after the small molecule Japans, which had started the field first until they ran into the issues, to go back and put effort into those Japans. That's what I'd recommend doing is nail down that PACAP is really important before we start going after the receptor for PACAP. And actually, I should not have said receptor. It's receptors. There's at least 4 different receptors, not even counting the splice variants of those receptors. So I think that Debbie's absolutely right that we need more information in the basic biology, which receptor to target from preclinical models before we start designing drugs for patient therapy.
Including the non-canonical receptors. So, I mean, I touched on the canonical ones earlier, but we mustn't forget there may be others. Still lots to learn about that, but we've got to keep an open mind. And I agree. Going after ligand is a great first go here.
Would you only go against PACAP, or would you also consider in the future going against BIP, for example? Tricky question.
So I always think of Star Wars. I like Star Wars a lot. And when Luke is going in to the Death Star to get the targeting that one little shoot down into the reactor core, and stay on target. So I would just stay on target on PACAP, show that works. And then I think that would be a reasonable target to look at. My hunch is it's not going to be very important, not as important as PACAP. So, but there's nothing, you got to do the experiments to see. We know that in mice and in humans, we thought for a long time that VIP, I like VIP better, just saying. It sounds more important than VIP. VIP, that it did not play a role in migraine. It's only until we realized, most likely because of its short half-life, that you need to alter the parameters. In mice, we need to measure behaviors right after injection of VIP. And in humans, Dr. Sheena had to do 2-hour-long infusion as opposed to 20-minute infusion in order to get a VIP response in migraine patients. So I think VIP won't be as strong of an inducer as PACAP.
Keep an open mind, ask the question, test the hypothesis. The biology will show us the way.
Thank you so much for joining this interview. I'm really proud that you have accepted. And thank you everyone for joining us in this interview. Thanks.